Good morning, everyone, and welcome to the Charlotte Medicine Friday morning meeting. My name is Lyle. I'm a fellow at Charlotte, and it's my pleasure to introduce my boss, Dr. Stacey, who's doing a talk today on the microbiome. The microbiome is obviously a very hot topic at the moment. We know that it plays a role in many parts of medicine, from infectious diseases to autoimmune diseases and malignancy. And so we're quite excited to hear some new information on the microbiome from Dr. Stacey. So at this point, I'll hand over to her to continue the talk.
Thanks very much, Lyle. So as you said, the microbiome over the last 10 or 15 years has really become a hot topic and a discussion in many areas of medicine, not only in infectious diseases, but as you said, in disciplines that treat autoimmune diseases, and obviously particularly in the GI area. And I think we're starting to realize that we are more and more closely connected to the environment than we thought, around us, within us, and we are much less the higher organisms than we think we are. We and the organisms that we carry are very integral parts involved in each other's survival. And of course, the organisms that we carry would be quite happy to find other modes of transport if we were not available, and they certainly do find other things to do when we expire on them. But we are less well equipped to do without them, and even poorly equipped to cope when the ratios of the organisms are slightly disturbed. So these days, you can't really be sure that any of the diseases that you treat aren't related in some way to function or dysfunction of the microbiome. So firstly, some definitions, just to remind everybody. I'm sure most people are familiar with the definitions, but just to remind people. The definition of the microbiome is basically a community of organisms, which includes bacteria, viruses, archaea, fungi, and their genes, which are present in a particular environment. And that term, the microbiome, incorporates the environment as well. And in our case, that environment is obviously the human body, but it could just as easily be a body of water or a spade full of sand. And these organisms may be commensal, they may be symbiotic, or they may even be pathogenic. And the balance between the helpful and the harmful organisms is generally maintained in a healthy person. The microbiota is the term used to describe only the organisms themselves. The metagenome is the collection of genomes and genes from the microbiota. The metatranscriptome is the collection of all expressed RNA in a specimen. And then the proteome is the entire protein complement in a sample. And the metabolome is the entire metabolite profile in a sample. And together, these give an idea of the regulation and the expression profile and the function of that microbiome. My slides don't want to change. All right, so these are the anatomical sites where it is generally very well accepted that a microbiome is present. Sites where organisms are expected to be and which engage in complex interactions with the host. The greatest number of organisms with the greatest diversity exists in the gastrointestinal tract. And of those, the most reside in the large bowel. And a disclaimer here is that a lot of the discussion today will focus on this large and important component of our overall microbiome. And other sites obviously include the skin, also a source of many and varied organisms, the orifices of the head, the nose, the ears, the mouth, the conjunctiva, the genitourinary system, the lung, and both the male and the female reproductive tracts. It's also, just to mention here, not true that we have 10 times as many bacterial cells in our bodies than we do human cells. It's probably closer to a 1-to-1 or a 1.3-to-1 kind of ratio, depending on how many times you've gone to the toilet in the last little while. But without doubt, we do carry a teeming civilization of organisms around with us. And one can never say never when it comes to a microbiome. It's an area that is always evolving, but it's probably fair to say that at present, there is little evidence for a microbiome in the brain, in the heart, the liver, the spleen, and the bloodstream. But there are researchers at the moment who have described what they believe is a microbiome in the brain. It starts to make you feel a little bit like a zombie, that maybe you are driven by your microorganisms inhabiting your brain. And the question always is whether or not the presence of of DNA that belongs to a microorganism is evidence of a microbiome, or whether that's evidence of infection. And a term that I came across that I particularly liked when I was reading is that infection does not a microbiome make. It is also possibly true that detection does not a microbiome make. And certainly if the detection of an organism is associated with pathology, then we would probably be more reluctant to say that this is a microbiome than that this is infection. And as I'll discuss a little bit later, one of the more recent microbiomes that has been postulated for the last sort of 8 or so years and is very hotly discussed and vigorously debated is the existence of a microbiome in the fetal environment. All right, so the first thing maybe we should talk about is how do you measure a microbiome? So the earliest microbiome studies involve traditional techniques that we generally use in microbiology. So the culture of a specimen to grow a colony of organisms. And this works very well for bacteria and fungi under normal conditions. Not so easy with viruses, not so easy to grow viruses. And certainly there are lots of viruses that are present in our microbiomes. And then obviously you identify the organism based on the morphology of the culture itself, what it looks like on the plate, what it looks like under the microscope. And then you can do biochemical tests to determine, which helps you to determine what that organism is based on that profile. So whether organisms possess particular enzymes that metabolize various substrates in various ways. And then you can say that this is most likely to be a particular organism. So the microbiome may only have become kind of, dinner table conversation in the last decade or so, but it's been of interest to some for much, much longer. So for example, Theodore Escherich, and you will recognize that name for it's obviously the E. coli has been named after him. So in 1885, he was already interested in what organisms existed in under normal circumstances. And he was culturing the meconium of newborns as early as 1885 to see what, if anything, grew in the first stools of life. Obviously, at that stage, he had only culture techniques available, and not everything grows as easily. In 1927 and 1934, there were 2 studies to examine the meconium of neonates. They used sterile nappies to collect meconium, and then they attempted to culture organisms from that meconium. And Escherich found that there was no viable bacteria, and the other 2 studies found that only about 2/3 of the samples didn't culture anything, and only about 1/3 of the samples actually cultured something. These days, obviously, culture-based technologies are not the only things we have available, and they are generally not used to study microbiota, and that's for several reasons. So the problems with culturing for microbiota studies are that many of the bacteria colonizing our guts are obligate anaerobes. And firstly, they often don't survive the procedures that are used for sampling the GIT, and they may also not survive transport to the laboratory and storage. Also, the bacteria that we find in the gut grow with variable efficiency in the laboratory. The organisms that would be most readily isolated would be those that grow under aerobic conditions. So these are obligate anaerobes. They don't really like to grow in oxygen. They will if they, some of them will if they absolutely have to. And some aerobic organisms sometimes will grow under anaerobic conditions, but they don't grow well. So the ones that we would be culturing are those that grow under aerobic conditions, those that grow at moderate temperatures, and those that grow with high nutrient growth media. And the bacteria that grow under those criteria probably only make up between 1% and 2% of the sample that you're actually interested in. So you're not really going to grow a proper panel of organisms that are actually present in the samples that you get. And essentially what you're growing is the weeds of the microbial world. So it can't really be said to represent the numbers and the variety of the microbes that constitute the gut microbiome. So really not a very useful tool for assessing the composition of the microbiome. So another way to describe the microbial population of a sample is to use PCR, which involves nucleic acid amplification of the whole 16S gene or portions of that gene. So the gene contains, sorry, I've obviously got my slides back to front, but the portion of that gene contains hypervarial regions by which you can speciate the organisms. This is done by techniques like gel electrophoresis, by fish flow cytometry, and by DNA microarrays to separate the fragments of different length. The problem with this technique is that it may not be sensitive enough to distinguish between some bacterial species, and sometimes even between genera of bacteria, or even families and orders of bacteria. So you can have a very general idea of what the orders or the families of the bacterial groups are, but not necessarily you can't necessarily speciate. Another problem with this method might be that bacterial species occurring in lower abundance may not amplify and then may not be detected. So by PCR, you need your primer to bind to a piece of bacterial DNA, and then to amplify that. And if there's very little of that DNA for the primer to bind to, and there's lots of other DNA that the primers may bind to, you may amplify those that are in abundance, but you may miss those that are in lower abundance. So this is what I meant by my slides being back to front. So the bacterial 16S ribosomal RNA, obviously all living cells contain ribosomes, and ribosomes obviously are the organelle that turn mRNA into protein. And ribosomes are composed of two subunits. One is a large subunit, one is a small subunit. And the small subunit contains an RNA molecule that is referred to by its size. So this would be either an 18S subunit in the case of eukaryotic cells, or a 16S subunit in the case of prokaryotic cells. The 16S subunit is encoded for by the bacterial genome, obviously. And it has both highly conserved sequences and hypervariable regions that can be used to then identify and classify the bacterial species. So current sequencing techniques have become really the gold standard for assessment of the microbiome. And current sequencing techniques allow sequencing of all DNA molecules that are present in a DNA mixture at the same time. So if you take a large mixture of DNA that would be contained, for example, in a stool sample, you can process that DNA on these very sophisticated platforms that allow you to identify all the DNA that is present in those samples. And that's in general what is looked at is the bacterial 16S ribosomal RNA sequences, and the identification of those hypervariable regions is what tells you exactly what species of organism that you are dealing with. And the reason obviously that we care about knowing what these organisms are is that we realize that the makeup of the microbiota has impacts on health and disease. And that influence starts very early in life. It starts at the very beginning of life. So the neonatal immune system is understandably immature. It has essentially no immunological memory, which is why neonates rely so much on the maternal antibodies, and to a large extent, the innate arm of the immune system for protection against infectious diseases in the very first months of life. Maternal antibodies wane over about 6 months. So it depends on what those antibodies are to. Some antibodies wane much earlier than others do. And so eventually, obviously, that child is going to need to mature its immune system in order to cope with challenges from infectious agents. So the components of the neonatal immune system that are functional are the following. So there are various cellular immune components. So we're talking about dendritic cells, neutrophils, macrophages, monocytes, and natural killer cells. And then there are some extracellular components of the innate immune system, which include complement, fibronectin, various antimicrobial peptides and proteins, and acute phase proteins as well. All right, so what is wrong with the neonatal immune system? So the neonate relies to a large extent on the innate immune system, but even the innate immune system doesn't function very well. So what is wrong with the neonatal immune system? So firstly, neutrophils are deficient in both quantity and quality. They have lower expression of toll-like receptor 4. They have deficient downstream signaling of MyD88. And ultimately what that means is that you end up with inhibition of TNF-alpha secretion. They have lower expression of adhesion molecules. So their neutrophils adhere poorly and they transmigrate poorly. They have deficient chemotaxis in response to chemotactic factors. So even if the neonate is secreting sufficient chemotactic factors, their neutrophils don't respond as well as they should be. They aren't as good as making neutrophil extracellular traps. So they're not very good at killing extracellular organisms as well. They have reduced phagocytic ability. So even if they can't phagocytose those extracellular organisms in order to kill them. And they have defective NADPH oxidase systems, so they have reduced generation of hydroxyl radicals. So they have neutrophils. Neutrophils are fewer in number, and there are many components of the activity of neutrophils that don't function as well as they should. What about antigen-presenting cells? So again, they have fewer dendritic cells and monocytes. The dendritic cells and monocytes expressed reduced levels of MHC class II molecules of CD80 and CD86. And basically what that means is that they are less efficient at activating antigen-specific T and B cell responses. The TEL-like receptors induce an anti-inflammatory response. So the anti-inflammatory response is production of IL-10, IL-6, and IL-23. This generally changes over the course of a year to a more adult pro-inflammatory IL-1 beta phenotype, but initially the phenotype is essentially anti-inflammatory. Natural killer cells, which play a major role in the resolution of severe acute respiratory viral infections like in influenza and respiratory syncytial virus in children, they do in fact have more NK cells than adults do. But they have a greater proportion of those NK cells are what's known as CD56 bright cells. So that's a subset that is in fact poorly cytotoxic. They express very high levels of cytokines and they home to lymph nodes. So they're not as effective as NK cells that you would expect to be in sites such as a mucosa, which would then encounter pathogens at the first stage. They have an increased expression of inhibitory receptors. So their receptors inhibit function rather than activate the cells. They have a reduced ability to lyse targets without undergoing differentiation. So an NK cell usually should be able to kill an organism without needing to undergo differentiation. And these NK cells in neonates don't do that very well. However, they do have similar antibody-dependent cytotoxicity activity to adult cells. All right, and gamma delta cells. So gamma delta cells represent about, these are gamma delta T cells represent 1 to 5% of the lymphocytes in peripheral blood of adults. And they are present in a lower percentage in human cord blood. Gamma delta T cells are one of the first responders to microbial infections like MTB, like Listeria and Brucella abortus. And the gamma delta T cells in cord blood have a naive phenotype, and they are poorly proliferative and they have reduced cytokine responses. So again, not cells that work very well. They are weakly cytolytic, and they have reduced perforin and granzyme B effector molecules. So what about the adaptive immune system? So the adaptive immune system in neonates also demonstrates both qualitative and quantitative deficiencies, and these persist till about 18 months. And this involves T cells and B cells. So what about T cells? So experimental studies of neonatal T cells have demonstrated polarization towards a Th2 response. So that's IL-4, IL-5, IL-10. They have decreased production of Th1 cytokines. So interferon gamma, IL-T, TNF-alpha. And the suppression of those, of secretions of interferon gamma and TNF-alpha by Th1 cells has been shown to be due to a higher expression and secretion of IL-4. It's also been demonstrated that there's a skewing of naive T cells towards a Th17 or a Treg lineage. And that's mutually exclusive. So you can either be a Th17 cell or you can be a Treg cell, and that there's a sort of reciprocal development pathway. Also, the presence of TGF-beta, along with IL-6, IL-21, drives naive T cells to become Th17 cells, whereas IL-2 induces TGF-beta treated naive cells to differentiate into FOXP3 Treg cells. And Treg cells, I'm sure you know, maintain immunological self-tolerance, and they negatively regulate various immune responses. So Tregs drive the immune response towards tolerance. Tregs are present in high frequency in cord blood and in neonatal lymph nodes. As I said, they are the reciprocal cell of Th17. Neonatal non-differentiated T cells will preferentially differentiate into Tregs when their T cell receptors are stimulated. CD8 T cells, they are reduced in number, and they have impaired CD8 activation, although they have an adult-like response to viral infections and vaccines. So the CD8 cells are less cytotoxic, and there are fewer of them. And then B cells obviously are naive. They've had no prior antigenic exposure, and they have a partially developed surface immunoglobulin repertoire. They have reduced strength of BCR signaling. They have higher expression of negative regulators again, and they have poor cross-linking of antigen to the B cell receptor, so that their responses to receptor signaling are poorer. And they also have defects in maturation and B cell isotype switching. So what this basically boils down to overall is a tolerogenic environment in the neonate, particularly at the mucosal site. Quite frankly, I think it's amazing that any of us make it out of infancy alive with immune systems that are so severely dysfunctional. And it's even more amazing that when we consider that we are immediately invaded by a multitude of organisms that colonize our skin and our mouths and our lungs and our gut pretty much before we've even let out our first scream. So basically what our immune systems at that stage show us are that there are highly regulated, predominantly anti-inflammatory innate phenotypes of neutrophils, antigen-presenting cells, NK cells, and gamma delta T cells. We have many neonates have many naive recent thymic immigrants. We have fewer effector memory T cells. We have a predilection for differentiation to Tregs. We have impaired Th1 cytokine secretion and reduced B cell receptor signaling. And what we need for our immune system to mature is we actually need the interaction with the organisms that we become colonized with in order to develop a mature immune system. So what happens if you don't get colonized with any organisms altogether? So if you prevent colonization of an animal altogether, so this is a kind of mouse in a bubble idea. And these are referred to as germ-free animals, which are bred and raised to be entirely sterile. And what happens is that these animals basically fail to develop normal immune function and immune anatomy at the mucosa and systemically. So firstly, they have grossly abnormal architecture of the gut-associated lymphoid tissue. They have abnormal architecture of the spleen and the thymus as well. And if we remember that the GALT is one of the largest organs of the immune system in the body, it includes the Peyer's patches, and the Peyer's patches house dendritic cells, neutrophils, macrophages. It includes the mesenteric lymph nodes, and it includes lymphocytes that are scattered throughout the intestinal epithelium as well. In fact, about 80% of the immune cells reside in the intestine of adults. Germ-free adults have germ-free animals, have a reduced frequency of CD4 and CD8 T cell subsets. They have reduced numbers of lymphocytes in the intestinal epithelium. They have altered T cell cytokine polarization, and they have a lack of diversity of alpha beta T cell receptors. If you introduce microbiota into the GIT of these germ-free mice, so if you give them normal mice microbiota, and even if you give them normal human microbiota, you can restore these defects. In particular, if you introduce Bacteroides fragilis, it's been shown to induce IL-10 producing Treg populations and to limit the pro-inflammatory IL-17 responses. Clostridial species also promote Treg development. And what's known as segmented filamentous bacteria, which are a form of gram-positive anaerobes, they promote the accumulation of Th17 and Th1 cells in the small intestine, and they drive the production of IgA. Just to mention here that I'm sure some of you are thinking, but Th17 are terrible pro-inflammatory cells, and they're related to all sorts of autoimmune diseases, and that has been found to be true. But in the situation in response to commensal organisms in the gut, IL-17 is related also to maintaining homeostasis in terms of preventing those organisms from invading. Because obviously many of the organisms that we regard as commensals do have the potential to cause disease, and we need them to stay in the lumen. We can't be entirely tolerogenic of those organisms. All right, so what happens to B cells in germ-free animals? Well, B cell maturation turns out to be normal in germ-free animals, but there are defects in the production of IgA and IgG1, both at the mucosa and systemically. And IgA functions to compartmentalize intestinal bacteria and to prevent the adherence of intestinal cells. So IgA is extremely important in the lumen of the gut. Dendritic cells, what they do is they will sample commensals in the lumen. They will then interact with T cells and B cells in the lymphoid tissue. And then the IgA producing B cells will migrate to the intestinal lamina propria and secrete IgA, which is then transcytosed across the epithelium. So there's an ongoing feedback loop with dendritic cell sampling and the production of IgA. So what about this newly described class of lymphoid cells that they're known as the innate lymphoid cells? So lymphoid cells are innate lymphoid cells are cells that have no T cell or B cell receptors. They remain cytotoxic and they remain immunoregulatory. So they mimic T cell cytokine secretion in an antigenic non-specific way. And there are a few groups. So there's group 1, 2, and 3. Group 1, which includes NK cells, are hyperactivated in germ-free animals. So these are cells that are responding to inflammatory signals from non-mucosal phagocytes, and they are hyperactivated. Normally, these inflammatory signals would be restricted in the presence of commensal bacteria. And then group 3 innate lymphoid cells secrete less IL-22. And IL-22 is a really important cytokine in the gut microbiome. It promotes the production of antimicrobial peptides. It stimulates regeneration of the epithelium. It regulates wound repair, and it increases mucus production. And if you are a germ-free animal, you secrete less IL-22, and so those functions are inhibited. Also, germ-free animals have abnormal intestinal epithelial cells. They have altered microvilli formation, and they have lower rates of cell turnover. So at acquiring your microbiome is extremely important. So how do we acquire our microbiome? So this is the traditional thinking. So the initial acquisition is either from, initial acquisition occurs at the time of birth is the traditional thinking. And the mode of delivery impacts the baseline composition of your microbiota. So during vaginal delivery, the neonate is more likely to acquire organisms like Bacteroides, Bifidobacterium, Lactobacillus, and Prevotella species. And during cesarean section, as the neonate isn't in direct contact with maternal, vaginal, or fecal microbes, is much more likely to be colonized by skin and environmental flora, including those from the hospital staff and from the hospital environment. And we know what kind of organisms we have in our hospital environment, so that's pretty scary. But the organisms are things like Propionibacteria, Corynebacteria, streptococcal species, and children born by cesarean section are also more likely to acquire a greater number of Clostridial species. Not all Clostridial species are terrible. Some of them are actually quite important in regulating immune responses. So very shortly after birth, the neonate will acquire organisms from the skin of the mother and from other individuals who may be handling the child, from the mother's breast, from the colostrum, and from the environment in general. The variation of organisms between cesarean and NVD babies decreases with time. And studies have shown that there's a variable duration of persistence of this difference. So it's been reported as anything between 1 month, so essentially only the neonatal period, all the way up to 7 years that this difference, this variability persists. The cesarean section may be associated with long-term health consequences, which may in turn, although this is not necessarily certain, be related to the different ways in which the microbiome is established. And these include altered cytokine expressions, susceptibility to immune disorders like asthma, allergy, type 1 diabetes, and obviously there's also this association with obesity. These associations do become quite hard to tease out because there are multiple other factors that influence the development and maturation of the microbiota. So subsequent feeding mode after delivery, diet, maternal age, the metabolic status of the mother, obviously your genetics, your lifestyle, and of course antibiotic usage as well during the lifestyle, that neonatal period. All right, but interestingly, recently, in the last 8 or so years, the idea of the sterile womb, which got its start in the days of Theodore Escherich, who was culturing meconium, the idea of the sterile womb has been challenged. And to some extent, so too the idea that the mode of delivery impacts us significantly on the composition of the microbiome. So this is still very, very controversial. And I don't really know which side of the fence I sit on personally. But with the advent of molecular techniques, much more sensitive techniques for developing and characterizing the microbiome, bacterial DNA has been detected in placental tissue. It's been detected in amniotic fluid. It's been detected in the umbilical cord of apparently healthy full-term pregnancies. So does that mean that there is in fact a microbiome associated in some way with the fetal environment? So if in utero colonization occurs, this would require dissemination of maternal microbes either by the ascending route from the production of the reproductive tract, from which we would expect organisms that we had mentioned earlier that you would acquire through normal vaginal delivery. So things like lactobacilli, Prevotella, etc. Or we might say that the microbiome of the placenta or the microbiome of the fetal environment is acquired by a hematogenous route from other maternal sites which harbor microbiota. So now we're talking specifically about the mouth and the GIT. So obviously what we're saying is that bacteremia would need to be occurring with viable commensal organisms from these sites. And this probably does happen in the normal course of our daily lives. So we do develop transient bacteremias with commensal organisms sometimes. So this might be from having a dental procedure, for example, or maybe even just brushing our teeth. So the idea that hematogenous seeding of a viable organism to the fetal environment might occur is not outside the realm of possibility. And also potentially immune cells might actually carry organisms from the mouth or the GIT to the placenta in order to facilitate the in utero establishment of a fetal microbiome. So when is the amniotic fluid positive for organisms? So most of the time, it is not possible to detect organisms by any method. So if you're using culture, if you're using PCR, if you're using sequencing, or if you're using a combination of those methods, 68% of the time, you don't find any evidence of bacterial organisms in the amniotic fluid. It is most likely, of course, to be positive under pathological conditions. So organisms are detected in 15% of cases of preterm labor, in 50% of cases of preterm premature rupture of membranes, which certainly sounds like it makes sense, in just under 10% of cases of preeclampsia, in 57% of cases with neonatal sepsis, which also tends to make sense and is probably sometimes related to that 50% of patients with bacteria in the amniotic fluid from preterm rupture of membranes, and in 6% of cases with small for gestational age pregnancies. And one wonders whether in those situations, whether those babies are not growing very well because that detection of organisms actually constitutes an infection rather than commensal organisms in the amniotic fluid. So the presence of organisms in the fetal environment may be associated with poor pregnancy outcomes. Also, the duration of labor is associated with the detection of organisms in meconium. So meconium, under 6 hours, if the mother has been in labor for only 6 hours, the meconium is usually sterile. If the mother has been in labor for over 6 hours, it's mostly culture positive, which suggests obviously ascending colonization given enough time. So are these organisms that are being detected necessarily commensals, or are they in fact invaders? So what about organisms in the placenta? So this is a study that really got people talking about a placental microbiome in about 2014. It was a study by Agard et al., which determined by metagenomic sequencing that a unique microbiome existed in the placenta, and it existed in the placenta in healthy, full-term and preterm pregnancies in babies that were delivered by cesarean section and in babies who had normal vaginal deliveries. And that the placental microbiome seemed to share a similar microbial pattern to the oral microbiota. But in this case, dominated more significantly by proteobacteria. So you can see that these colors here, these are the proteobacteria from the placenta. You can see that they are present in the oral mucosa as well, the tongue, the supragingival plaques, and the buccal mucosa, but significantly more in the placenta. So dominated by the proteobacteria, and also including a phylum of bacteria not found at any of the other sites, and those are the tenericutes. And that phylum includes traditional pathogens, ureaplasma and mycoplasma, that we know are associated with the uterine environment, and also we know are associated with preterm delivery and abortion. So whether or not the detection of that particular group of organisms means that they are part of the placental microbiome, or whether they are actually pathogens. The other thing is that the placental microbiome did not appear to be similar to the vaginal flora that we would expect. If the placenta was delivered through the vagina, we would expect that the placenta would become colonized during that process with vaginal flora. And we can see that the pattern here, this is the posterior fornix of the vagina. It's mostly firmicutes. and it's not similar to the pattern that was observed in this study in the placenta. Organisms in this study were detected in low abundance. They were detected more often in preterm spontaneous births, which one would call an adverse pregnancy outcome, and also more frequently in association with a remote history of infection during pregnancy. And by this, they mean things like UTIs in the first trimester. So the presence of infection is associated with the presence of organisms in the placenta, and also may add the potential confounder of antibiotic exposure if those infections were treated. This bubble plot on the right here, where's my pointer gone? This bubble plot on the right here shows that the most prevalent single species in the placenta in this study was E. coli. So there's the largest dot. So the most prevalent organism in the placenta in this study was E. coli. And I must confess that my gut response, pardon the pun, is that this is quite suspicious for a contaminated specimen or otherwise inaccurate results. And I don't really have any particular evidence to support that reaction that I have. But I feel that a potentially pathogenic organism like E. coli, which, you know, for an organism like E. coli, which we know has significant pathogenic potential, to take up residence in an organ like the placenta, even if we're calling the placenta a very tolerogenic organism, to have no inflammatory reaction to the presence of E. coli, essentially, you know, in an internal organism, makes me a little bit uneasy. So what are the reasons that you might treat the existence of a placental microbiome with some caution still? So firstly, the technology for the detection of organisms of the microbiota is really very, very good. So the sequencing techniques that are used to detect organisms in the microbiota and to speciate organisms in the microbiota are really very, very sensitive. But you might only expect 0.002 milligrams of bacterial DNA to come out of each gram of placental tissue, which means that you are dealing with a very, very low biomass of organisms present in that tissue. And so that increases the chances that the bacterial sequences that you are detecting are actually coming from somewhere other than the placenta. So it's really, really hard to collect a completely sterile sample in a clinical setting. So even if you're cleaning with your alcohol, or you're cleaning with your betadine or whatever, it's actually very difficult to collect a completely sterile sample. Also, you run the risk of using reagents or extraction kits or fluids that may be contaminated by DNA from somewhere else. And this is referred to as the kitome, you know, microbiome kitome. It becomes very important to decide whether or not your kit might be contaminated with DNA when you are dealing with a very, very low biomass in a sample. Similarly, there's something that's known as the splashome, which refers to the possibility of splashes from one well of your plate to another, for example, when you are preparing your samples. And several studies, including this sort of seminal AGARD study, shows that there's an overlap between the genera described in the placenta and genera found in common water and soil samples. So the question is, are these studies just demonstrating picking up some kind of contamination and not actually demonstrating a placental microbiome? Or possibly picking up fragments of organisms that have lodged in the placenta but do not in fact reflect viable organism as part of a microbiome. The other question is that it is possible to deliver germ-free animals from non-germ-free mothers by cesarean section into a sterile environment and keep them sterile by raising them in that sterile environment. So if you can deliver a germ-free animal from a non-germ-free mother, how is it possible that there is a placental microbiome establishing the microbiome of the child during gestation? So really, there are more questions than answers when it comes to this. It's an interesting area to watch. At the moment, the evidence is kind of balanced. The experts are hotly debating it. I'm probably falling in the direction of the sterile womb hypothesis, but I would be equally happy to be convinced otherwise. All right, so while it might be as yet unclear how exactly the colonization of the human host by various microbiota starts, and we don't know how early it starts, it's probably still fair to say that you get your biggest dose of microbes soon after you are born. And we realize that colonization is essential for effective maturation of the immune system. The immune system and the microbiome develop in parallel in very early life. So what do you need your microbiome to educate your immune system to do? So secretion of antimicrobial peptides by neutrophils and intestinal epithelial cells is attributed to gene induction by gut commensals. Maturation of Th2 dominance is driven by bacterial interactions, which help to promote that barrier function that is essential to prevent invasion from commensal organisms and to regulate the Treg and T helper cell balance. Bacterial products, specifically polysaccharide A, which comes from Bacteroides fragilis, and short-chain fatty acids, or particular species like Clostridia, induce functional Tregs in the colonic lamina propria. This is related to protection against immune-related disease, and it's also necessary for tolerance to food antigens. An increased abundance and diversity of Bacteroidetes is associated with improved Th1 responses in neonates. So while a neonate is essentially a tolerogenic organism immediately after delivery, the neonate does need to defend itself in some way against pathogenic invasion, and the Th1 responses are required for that. Segmented filamentous bacteria attach and actually penetrate into the epithelium of the terminal ileum, and that induces Th17 cells, which again are important for protection against invasion by commensal organisms. If you have insufficient microbial stimulation from your commensal organisms, this results in class switching of your mucosal B cells to IgE rather than IgA, which is what you want to protect your mucosa. And it results in increased serum IgE levels as well. The antigens and metabolites of the microbiota promote plasma cell differentiation in the mucosa and systemically. So that that means that your plasma cells can produce more memory-like profile in terms of your antibodies. As I mentioned earlier, there's a positive loop of IgA secretion. So there's the IgA will coat and uptake some of the luminal take up some of the luminal microbes. They will transport them for processing in the Peyer's patches, and that reinforces the antigen-specific responses and increases the secretion of IgA in response to your commensal organisms. And IgA secretion as well modulates the gut microbiota composition. Segmented filamentous bacteria also stimulate T-cell independent IgA production in the neonate. And then just to say that the microbiome remains unstable in humans for about 2 or 3 years. So in the same way that you can reconstitute the immune development of a germ-free animal by introducing microbiota, you can restore or modulate the composition of the human microbiome potentially within that 2 to 3 years while the microbiome remains unstable to normalize immune development. The same, however, is not true in adults. So there's this critical window early in life where the establishment of your microbiome is going to impact your immune responses long-term for the rest of your life. And once you become an adult, you are not able to do that anymore. All right, so really what I'm trying to say with this rather sort of disparate presentation is to love your microbiome. Try not to kill it too much with antibiotics, and the same applies to your patients, obviously. Treat your microbiome with love and respect, because these bugs are essentially keeping you alive. Thanks, anyone. Any questions?